Good afternoon, virtual audience. We have heard great science today, and this is a very exciting symposium. And now we're going to move more into cellular and molecular biology. And we are interested in infections because they represent one of the major risk factors for cancer. And we are particularly interested in bacterial infections. Now, we know that viruses cause cancer, papilloma, hepatitis C, EBV, but there are also strong epidemiological evidences that bacterial infections are also associated with cancer. But if viruses can put their genetic material inside the nucleus and drive somehow cellular transformation, which are the mechanisms that bacteria can use to increase the chance of cancer development? This is still a big unresolved question. And most of our knowledge comes from studies of cancers in the gastrointestinal tract. Now, besides few exceptions, most of the cancers that are associated with bacterial infections are adenocarcinomas. That means cancer of epithelial cells. So we can simplify in very lay terms the two negative outcomes of a bacterial infection. So rather the epithelium succumb and there is an ulcer, or the epithelium resist, but at the cost of an increased risk of developing cancer. Now, if you look at the schematic, which represents the host-pathogen interaction, at least from our point of view, you can see that infection, inflammation, immune response, mutations, and cancer indeed are converging to the same place, which is the epithelial barrier. So this means that if we want to understand infections and cancer, we need a good epithelial model to make observations and test hypotheses. And obviously, organoids represent a formidable tool to understand epithelial biology. They are made of polarized cells with an apical and a basal side. This is just to show that the epithelium is the center of our attention, the center of our study, because most of the events that we are studying are happening here. And yes, organoids. If we want to understand, we're talking about organoids. So the features of organs are that they are made of polarized cells with an apical and a basal side. This culture is stem cell driven. So this means that we can regenerate healthy or diseased cells in vitro for very long. They are capable of multi-lineage differentiation. That means that it is possible to derive different type of cells within the epithelial lineage, and they can be derived from human, which is an advantage if we want to understand human pathogens and human cancer. So this is beneficial for medicine, but is also a great biological challenge. And they are also a starting point to develop more complex cultivation system, as we will see in this talk. So is it progressing? So now you should see a human body showing our main epithelial models. Thank you. So we derived cells from the stomach to study infection with Helicobacter pylori and its impact on gastric carcinogenesis. And then we also study the human gallbladder to study the impact of typhoidal Salmonella in the development of gallbladder adenocarcinoma. So in this part of the talk, I will talk about the generation of this model, which derives from organoids, and which we think is more suitable to study infections. We call this model the mucosoids culture because this culture model has most of the features of a real mucosa, including mucus production. So we developed these for the first time from the stomach. We start from lead resections of the stomach. These are obese patients that are undergoing reduction of the volume of the stomach. We can obviously from the epithelium derive organoids, but then we took these organoids, we dissociated this in single cells, and we seeded them on regular transwells in air liquid interface, as you can see from the schematic here. So this is a very simple culture system indeed. And then you can cut the filter out and use it as an histological sample. So this is what is happening when we seed the cells. So one day post seeding the cells, the epithelial cells are quite flat and unpolarized, but with the time passing and with the starting of the air liquid interface, they become taller and polarized. And polarization is here shown by the ecadherin, which is basal and lateral, but we will see later in a much better detail. But the good thing about this cultivation system is that, as it has happened with organoids, we are able to propagate this. So we basically trypsinize the cells, and then we see them with a split ratio from 1:3 to 1:5, and we can run this culture for years. So this is about polarization. So ecadherin is basolateral, and actually basically the top part of the filter of this insert represents the apical side. And whatever we put here in the cultivation medium will go indeed to the basal side, because the filter is porous, so it will go this side. And also, as you can appreciate, there is all the tight junctions are decorated here. So this is an anti-occludin antibody, just showing the cells are bound with tight junctions. The other feature is mucus production. So these cells here, they start producing mucus, like the epithelium in our body, and this is why we call them mucosoids, because we think that this is one of the main feature which the mucosoids are representing from the mucosa. So cells in the mucus are proliferating, or at least there are some spots, some foci of proliferating cells. And here I'm just showing a bunch of markers which are constant each passage. So each passage is 1 month of culture. So here it's 8 months represented. And most of the markers we have tested, epithelial markers or even lineage specific markers, because there are different cells that are different type of epithelial cells populating these mucosoids. And also some stem cell markers are constant over time. So this is how the stomach mucosa looks like. And we really wanted to explore here the multi-lineage differentiation capacity. So how we can obtain the different kind of cells. So the stomach is organized in invagination called gland, and there is a monolayer of cells. I mean, you can appreciate here the single cells that are invaginating down. I mean, this is another schematic that shows how the gland should look like. And there are different cell types here. So there are the one producing mucus, the one producing digestive enzymes, the one producing gastric acid, and the one producing hormones that are regulating all these other kind of cells. So we wanted to explore to make sure that our model is a truly representative of the stomach, if we could differentiate all the cell types. And also if at the barrier level it looks similar. So this is an immunofluorescence of the same stomach. And as you can see, this mucosoid culture, they really look very similar in terms of barrier structure. So this is about the different cell types. Again, cell producing mucus, like in the first two here, cell producing digestive enzymes called Schiff cells, and also parietal cells. So to cut a long story short, basically, we have found that a combination of activation of the EGFR signaling pathway and of the BMP bone morphogenetic factor signaling pathway. Here you can see the different combination. We can basically get most of the cell types that are populating the human gastric gland. So these are normal epithelial cells from normal stomachs. And also we went into the function. So when we analyzed the differentiation into the mucus producing cells, of course, we could see the mucus. We did mass spectrometry, and we could find the typical factors which are present in the mucus. We also analyzed digestive granules. So these are zymogenic granules, and we could find the precursor of pepsin inside these granules. And also we analyzed gastric acid production. So in this experiment, we could see by electron microscopy that there are all these typical canalicula, which are bringing the gastric acid to the lumen of the stomach. And also when we provided the hormone, which stimulates gastric acid production, and a dye, which measure basically lowering of the pH, we could see that there were many cells over time producing gastric acid. So basically, we are quite convinced that this model is the highest representative of the gastric barrier in the body. So how we can use this to understand infection? So we have tried infection with Helicobacter pylori, and here are just some electron microscopy pictures that have more of an artistic value rather than scientific, but still you can see that the helical shape of Helicobacter is lost, and this is a symptom of bacterial stress. They're also vesiculating a lot here. So probably it suggests that there is some kind of response from the epithelium to the bacteria, which we will explore later. But eventually some bacteria can land intact on the epithelium. And I think that this is when host pathogen interaction starts. I mean, when bacteria can land on epithelial cells. But Helicobacter is a bad pathogen. It can go even further, and it can cross the epithelial barrier. So it has a protease which is able to open tight junction, and it migrates to the basolateral side. So what we could understand by using the mucosoid culture, here is one experiment as an example, is that different kind of cells at different position might have different capacity of responding to infection. So it has been demonstrated in the past that gastritis, which is the typical disease that arises after Helicobacter pylori infection, happens only when Helicobacter goes inside the gastric gland. So it's not just swimming on the surface, but it goes inside the gland. So we did analyze what is the difference between the response of the cells inside the gland versus the response of cells which are here at the surface. And we used our differentiation protocol. And in particular, we used this factors, Wnt and responding, which promotes this lineage versus this lineage to understand the response against Helicobacter. So we have infected both treated and non-treated cells, and then we have run a microarray, and we understood that basically mostly all of the NF-kappa B target genes, which is a master mediator of inflammation, were present only when Wnt responding are there, meaning that only these cells are probably able to start a proper inflammatory response. And this makes sense because 50% of the human population has Helicobacter in their stomach. Well, we don't see gastritis in 50% of the population. So as long as it stays here on the lumen of the stomach, probably inflammation doesn't start. Another interesting experiment that we've done is to follow the infection over time. And this is 1 month. And we have been measuring by qPCR the amount of H. pylori on the mucosoids. And what we could see is that there is a decrease of the amount of bacteria on the mucosoid. And this is quite surprising because if you try to infect a cell line with H. pylori, well, the cell line will die, not the bacteria. So this is again suggesting us that there is an epithelial response against infection. And we wanted to understand what is this about. And we think that this is very important also to understand antimicrobial resistance. And so how can bacteria so the question is, how can bacteria evade the humoral response, which is quite potent, but somehow it doesn't work. So we try to understand whether there are some properties in the mucus that are important for fighting pathogens. So this is a mucosoid section which is unstained and also still under the wax, so not even dewaxed. And here would be the epithelial layer, as you have seen before. And here there is another important layer, which is probably the forefront of our innate immunity. So this is the inner mucus layer. So mucins, before they get hydrated and they become the gel-like mucus that we are used to see, they form a stiff layer, which, as we knew, it blocks bacteria, and we could mimic this in vitro as well. So most of the bacteria are blocked at the inner mucus layer interface. But some bacteria can make through this layer and they can land on the epithelium and make a colony. So the mucus is for sure a passive barrier. So it's a wall basically against bacteria before the epithelium. Then we wanted to understand if there are features inside the mucus that can somehow counteract infection. So we have taken the mucus out of the mucosoids. So as you can see in this diagram, we incubated Helicobacter inside, and then we plated after 1 and 2 hours. And with our surprise, we couldn't see any effect of the mucus on H. pylori. So there is nothing in the mucus that counteract the bugs. But if we take the mucus from a previously infected culture, and then we incubate pylori in this mucus, then none of the bacteria survives. So this means that somehow the epithelium must be triggered. So there must be some information that the epithelium gets in order to secrete some substances, probably antimicrobial peptides in the mucus, with the effect of killing bacteria in 2 hours. So that might explain why we have seen this decrease in bacterial load over time, because the mucus is probably toxic. But the bacteria might have developed some mechanism to evade this. And this is exactly what I would like to show in this slide. This is an experiment in which we contributed with our model. And it comes again into a topic which has been mentioned today, which is lipids and interferon signaling. So to cut a long story short, Helicobacter pylori is auxotroph for cholesterol. So to survive, it needs to take cholesterol from somewhere else. And one place which is quite rich in cholesterol is the plasma membrane, and specifically the cholesterol-enriched domains, which are also known as lipid rafts. On the lipid rafts, there are many important receptors, among which the interferon gamma receptor and IL-22 receptor. So basically, when bacteria lands on epithelial cells, they glycosylate cholesterol, they extract cholesterol, they disrupt the lipid raft, and interferon gamma and IL-22 receptor are not functioning anymore. And we could beautifully demonstrate this by using the mucosoid model. So this is a view from top. And we were measuring phosphorylation of STAT1 and also of STAT3 after infection. So in red, you can see the infection. And basically, all the infected areas do not show phospho-STAT1 after interferon treatment. So this means that these cells are not sensitive anymore to interferon gamma and also to IL-22. So this means that when the immune system comes close to the epithelium and tells the epithelium, maybe with the interferon gamma, to do some response against the infection, the infected cells cannot reply to this stimulus. And maybe this might be one mechanism that Helicobacter can use to escape host response, to escape a humoral response, but also adaptive immune response. So to summarize this part of the talk, I mean, we have been developing this tool, which we call the mucosoids, to understand infections. And also we were able to differentiate all the cell types into the different kind of cells of the stomach. And we have published this recently in gastroenterology. And also we could use this model to understand how bacteria can escape the epithelial response to infection. In the second part of this talk, I would like to talk about another bacterial infection, which is associated with cancer, that is typhoidal salmonella, and specifically Salmonella typhi and paratyphi A. So this is quite an exotic disease, but also very interesting from our point of view, because basically the area where typhoidal salmonella, so these are very specific strains of salmonella which are expressing the typhoid toxin. These strains are endemic in the northern part of India and in Chile. These are exactly the two parts of the world where carcinoma of the gallbladder is more frequent. And in fact, carriage of typhi and paratyphi A, so chronic carriers, have a higher chance to develop carcinoma of the gallbladder. And actually, this was understood for the first time in this beautiful epidemiological study that was done here in England. And in particular, this is a nice story. So there was a cargo of contaminated meat that landed in Aberdeen. So the meat was from South America. There was an outbreak of typhoid fever, and 25 years later, there was a number of cases of gallbladder adenocarcinoma. And this association was found again also in Chile and in India, demonstrating that there is a strong epidemiological evidence that associate chronic carriers of Salmonella typhi and paratyphi with the onset of gallbladder adenocarcinoma. So how Salmonella ends up in the gallbladder, I mean, it's still not known. It might come from the liver or maybe retromigrate from the colon. But for sure, what it does is that it starts coating the gallstones. So usually the patients with gallbladder carcinoma do have gallstones which are coated with biofilms of Salmonella. And eventually the Salmonella can be shed and go into the epithelial cells of the gallbladder. And we think that this is very interesting because if we want to understand the association between bacterial infection and cancer, I think we should look at the hallmark of cancers, which were published by Hanahan. So bacteria can contribute to many of these, but if we want to look at direct impact of bacteria into cancer, I think we should look at genome instability and mutation. And that's why we got interested in this bug, because the typhoid toxin, which is expressed specifically by this strain, there are also other bacteria which are producing this kind of toxins. It's a DNase. So basically, this toxin goes into the nucleus, and there is this subunit called CTTB that is an homologous of DNase 1. So it goes into the nucleus and it cuts DNA. And this toxin is usually secreted once Salmonella is internalized in cells. So to study this, we have developed organoids of the gallbladder. These can be propagated for years. The gallbladder organoids are very similar to the epithelium of the gallbladder in situ, as you can see from these pictures. So cytokeratin 19 is an epithelial marker, tight junctions, production of mucus, and also they are stable over time. And I'm not going into these details, just trust me. And also another nice feature, they are able to transport bile. So we have used a surrogate of bile, which is green. So this is a dye which is chemically similar to bile salts. And the gallbladder organoids are able to pump in the bile. If we block the drug, if we block the channel, which perform this function, we don't get internalization of this fluorophore anymore. And also, if we use stomach organoids, which do not express this pump, of course, they do not internalize this surrogate of bile. So we have used this model to study Salmonella infection. We have been infecting organoids. We have been producing mucosoids of the gallbladder. And also to study the intoxication, we have used just normal cells of the gallbladder plated on plastic to see the short-term effect of the toxin. So results were quite nice. So we have been producing a mutant of the Salmonella, which lacks the subunit which performs the DNase activity. And we have been measuring the amount of damage by using this marker, which is gamma H2X. So this is an histone variant which is recruited where the DNA is broken at where there are double-stranded breaks. As you can see here in this panel, around the Salmonella infection, there are many foci of gamma H2X, which suggests that there is lots of genomic instability which is going on around the infected site. When we infected with a mutant, we didn't see this effect. And we could quantify, so this is an infection with organoids, we could quantify this effect in this graph. So basically, the first 3 rings of cells around the infected cells have a higher chance to experience genomic instability. So this means that this effect of genomic instability, which is caused by the infection, is paracrine. So it's also extended to the bystander cells. To understand the role of the toxin, we also perform infections of mucosoids. So with mucosoids, we can extend the infection further up to a week in this case. And what we could see is that we would have expected typhoid toxin to block the cell cycle, because if we think like in the textbook, there's toxins are causing DNA damage, and DNA damage to be repaired needs the cell cycle to be arrested. But actually, what we could see is that the cell cycle arrest does not depend on the toxin. I mean, there is, yes, cell cycle arrest at 2 days and also at 7 days. So here we have done a microarray in which we have measured all cell cycle related genes divided by a phase of the cell cycle. And then we could see that in any case, regardless of the presence of an active or a non-active toxin, there is an arrest of the cell cycle. So the cell cycle arrest does not depend on the toxin. So to understand what is the mere effect of the toxin, what we have done was to take cells, we put them in plastic, and then we just took the toxin out of the culture. So we cultivated bacteria with a minimal medium, we filter it, and then we have intoxicated the cells. Again, we could confirm that there is damage in these intoxicated cells. So this is the wild type versus the knockout supernatant. So there are no living bacteria here. And then we compare this to etoposide. Etoposide is a topoisomerase inhibitor. So when filaments of DNA are crossing within each other, they are reassembled back. But if you put etoposide, this doesn't happen and it generates a break. So the amount of damage that we could see with the toxin is similar to what etoposide can cause. And we could confirm this also by another assay. This is called COMET assay. So cells, while migrating in a gel, if they have broken DNA, they leave a longer tail. And the tail was longer in the cells which were intoxicated with wild-type toxin. So with our great surprise, when we measure the proliferation by this marker Ki67, we could see no difference in proliferation between the wild type intoxicated versus the knockout or the sterile medium. While when we treated them with the topocyte, there was a brutal arrest of the cell cycle. And actually, even more interestingly, we could see that the fraction of cells which are proliferating, so the Ki67 positive, are also the one which are experiencing the higher amount of damage. So basically, these cells are proliferating and they're also experiencing damage at the same time, and they do not stop their cell cycle to repair the damage. So we think that this is a starting point that puts the cell at risk of cellular transformation, and we have published these results last year in Bio. We have seen a similar mechanism also for other bacteria and other toxins. So there is another bacterium which is very well known. It's Escherichia coli, and some strains they do produce a toxin which is encoded by this island PKS, and the toxin is called Helicobacter. So we have developed mucosoids from the colon, and then we have infected these mucosoids of the colon with Escherichia coli, which produces the Helicobacter. And again, we could see that there are cells that are Ki67 positive. That means that they are proliferating and they're also experiencing damage at the same time. So this might be a feature of many types of infections with this toxin. So these toxins are not really provoking cell cycle arrest as it was thought before, but maybe at low doses, they are still able to cause damage, and at the same time, the cell cycle doesn't stop. So the cells are keeping proliferating. So in conclusion, we have been generating this mucosoid culture from the stomach, from the gallbladder, from the colon, and we could demonstrate that we can get lots of insights into epithelial host response to infection and into carcinogenesis. But in theory, this cultivation system can be used for every type of a simple columnar epithelium. We have tried, for example, also fallopian tubes, and here you can see the typical cilia of the fallopian tube, so the one transporting basically the oocytes. And we have patented this, and we will hope we can exploit this further. And what we are doing next is to make even more complex models, so to extend, so to make these models more three-dimensional. So instead of having a flat layer of epithelial cells which mimics the barrier, we would like to fold this barrier to make the real niches that are found in the gastrointestinal tract, so glands and crypts, and of course to do infection studies with this. And this is my thank you slide. So thank you for the attention, and also thanks to all the collaborators that have been participating in all these studies. I mean, here in this slide is just the main people, but there are many more from the Max Planck Infection Biology, in which I did my postdoc, and especially I would like to thank Thomas Meyer for being my mentor over many years, and then also all the collaborators, the clinical collaborators in various hospitals in Germany and here also in Oxford, and of course my current lab members, which are supporting me to go on with this exciting research. Thank you very much.